today we are going to show you a lecture video about DNA replication, transcription, and translation. Hope you like it!
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases represented by C, A, G, T. The 2 strands are complementary. This means that wherever there's a T in one strand, that would be an A in the opposite strand. And wherever there's a C, there would be G on the other strand. Each strand has a 5' end and a 3' end. The 2 strands running in an opposite direction. This determines how each strand of DNA replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide the template for creating a new strand in the DNA. An enzyme called primase stops the process. This enzyme makes a small piece of RNA called the primen. This marks the starting point for the construction of the new strand DNA. An enzyme called DNA polymerase binds to the primen and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction from the primen to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' end to the 3' end direction.
The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase cannot fully make this strand in a series of chunks called Okazaki fragment. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in a 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made. The other strand, the new DNA has been made. The enzyme exonuclease removes all the RNA primens from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous
DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is a TTA box. An enhancer region may also be involved. Several complexes known as transcription factors are required for successful transcription. The first is TFIID, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the TTA box. To position TTFIID, new transcription, and initiation site. Other transcriptions.
This complex prepares the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
Polymerase dissociates and the newly formed strand of RNA is released.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunit of ribosome, tRNA, and finally the release factor. This process has 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' called a poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5'. Translation initiation begins when the small subunit of ribosome attaches to the cap and moves to the translation initiation site.
Site.
tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first codon is typically AUG. Attached to the end of tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon.
Responds to the AUG codon.
The large subunit now binds to create the peptide or P site and the amino acid or A site. The first RNA occupies the P site, the second tRNA enters the A site, and is complementary to the second mRNA codon. The methionine is transferred to the A site amino acid the first tRNA exists. The ribosome moves along the tRNA and the next tRNA enters.
These
are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA. The ribosome moves along mRNA and new tRNA enters. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and a newly formed protein is released.
So guys, did you enjoy watching our lecture video?
Hope you do!
Please subscribe to our video and leave a comment below.
And share it too!
Thank you. Goodbye. Goodbye.
Take a sip from a secret